Hi, my name is Alexa Alvarez, and today we are continuing on our Lab 5, which is Bacterial Morphology and Staining Techniques. It's my little card. So the premise of today's experiment was for us to get an idea of what different bacterial cells look like under different staining conditions. So we worked with our we worked with our collecting dental plaque essentially, and we did a direct stain and a negative stain. So we're able to see how differently each particular cell stains within that particular culture. And we also worked on doing differential staining, and on the differential staining, I feel like was probably the most intensive in terms of procedures and I want to say kind of outcome. Based on the imaging that was shown on the HOL site, I don't know that I received, or I don't know that my stains actually picked up as well as what was expected. So if I hold this up against something white, there really isn't anything there. There's a little bit of a violet stain, but like this one, hardly anything. The only one that I picked up some was yeast, and it mostly received, mostly absorbed some of the violet stain. I didn't really see anything else. So I'm not entirely sure exactly how or what I was to do to get my stains to look a little bit more vibrant. Other than that, I was looking at, when I was doing the microscopy on it, so I was looking through the microscope and I was able to see on the yeast. It was very easy to find. There was a lot of cells. So I'm kind of wondering if on these, if maybe putting them in the refrigerator, because I believe I had read instructions that putting them in the refrigerator was supposed to preserve them, but being that they're technically, there's cultures in there, I don't know if the change in temperature maybe stunt the bacteria on the epidermis and the E. coli, or if maybe I didn't pick up enough of the specimen within these. My yeast looks, there's a lot of turbidity in it, and I figured I would get pretty decent results out of that one. But I'm not entirely sure what all happened with the other two. Looking at the microscope for the other two that didn't receive too much of the stains, if at all, the bacteria was very sparse. So that's why I'm thinking I probably didn't pick up enough, or maybe, I don't want to say it dissolved in there, but maybe I needed to kind of try and get way deeper in there and pick up more of the substance of the specimen versus the liquid. Also on the rinsing process, I feel like maybe the water dilutes some of the dye. I know on the first experiment we were supposed to kind of blot it dry, but on this particular experiment it didn't ask us to do so. So I attempted to kind of absorb as much of the water as possible, but within the center there was still some residual water left over in there when I would have to add the other dyes. Other than that, I think everything went well. I was still able to get some decent images out of each one. I looking at the text and looking at kind of everything we were doing in terms of leading up until the last of the assignment, I think I have to read up a little bit more on the gram-positive and gram-negative, because I think I may have them swapped in my mind. And based off of what we read, a gram-positive was supposed to absorb most of the dye, but then the negative was not. But now that I was kind of going through everything, being that a gram-positive has a thicker outer cell wall, it's not supposed to. So I think I need to work on that a little bit more to see, just to kind of clarify on my end to make sure that I actually understand what all is going on with bacteria. Other than that, I think that's all I have for now. Thank you so much.